Perfect. So let's hope my computer doesn't have a stroke again. But yeah, so this is a recap of part 2 of lecture 24. So this one, I believe, was on signaling between organisms, whereas before we talked about defense mechanisms. So yeah, so basically let's talk about signals and what they even are in the first place, right? So these are molecules that work in the body to control different things like growth, development, metabolism, and other biochemical reactions. So they can regulate the speed at which these things happen, and they can also stop and start these processes, depending on what the body needs and what some of the signals they're getting from the environment. So basically, what types of signals have you learned about already, like I guess in lecture? So you've learned about intra-organismal and inter-organismal signals. So intra, whenever you see that prefix, it means within. So this is talking about signaling within an individual. So maybe from one part of the body to another part of the body. And then you also talked a little bit about inter-organismal signaling. So whenever you see the prefix inter, you want to think between things. So between two different organisms, right? So this can either be between organisms in the same species or between organisms in different species. But the point is, we're not talking about just signaling within one individual. And then there are also environment to organism signals. So this is kind of getting an input from the environment using different sensory mechanisms, and then taking that in and understanding how to respond properly according to what that signal was. So a good example of this that we've already talked about were the meristems that we found in plants. So I said before that meristems are very similar to stem cells, except it's kind of like the plant equivalent. And I said that they have apical meristems and lateral meristems, right? So the apical meristems are the ones that enable vertical growth. So it allows the plant to grow taller. So this would be done maybe in order to outcompete other neighboring plants for access to sunlight, right? For photosynthesis. So maybe the environmental signal would be that they're not getting enough light. So they have systems in place that can recognize this signal and then appropriately stimulate these meristems to grow, to allow the plant to grow taller so that it can basically outcompete the plants next to it and then have more access to sunlight. So that's just one example, but we'll talk about a few more. So this is kind of the signaling that we talked about last semester with Dr. Durnford. So this was kind of the signal transduction cascade, and we went into a lot of detail about some of the receptors. Remember, we talked about GPCRs, and we also talked about RTKs. Lucky for you, if that freaks you out, this semester isn't that in depth, and it's mostly about the general concept of signaling and how that works in an ecosystem as opposed to signaling within one individual. So just as a refresher, though, remember that there were 3 steps for virtually every signal transduction cascade. So there is the reception, so that's actually getting the signal. So that would be like the signaling molecule, whether that's a hormone or something else, binding to the receptor, either on the cell wall, like we talked about last semester, or sometimes in the cytoplasm, which we'll talk about this evening. And then the second step is transduction. So that's kind of playing telephone inside of the cytoplasm. So basically the message is passed on between molecule and molecule and molecule. And then lastly, it's delivered to a final molecule that can actually elicit the response that's needed and requested by the signal. So let's say it was some sort of kinase that was the last one in the chain, and it went to go to the nucleus, and it phosphorylated a transcription factor and then began the transcription of a certain gene. So maybe that was the request of the signal in the first place. But the point is, it really does come down to 3 basic steps, although those steps will definitely vary depending on the signal and also depending on the receptor. But again, you don't really need to worry too much about it. Just kind of recognize that there are 3 steps in the process. Right, so what are, do you remember some of the many types of signals and pathways that we talked about last semester? I'm not going to go into too much detail because, again, it's not as important this semester, but we did talk about signaling between different cells. So we said that it can be a short distance or local signaling, or it can be long distance signaling. So you can have paracrine signaling, which means that the cells have like basically they need to be really close together. You have synaptic signaling, which is what happens in your neurons. You have cell-to-cell recognition, where you have a receptor on one cell and then a signaling molecule or whatever on the other cell, like a spike protein or something. And then in long distance, we talked about hormones, and that's the one that we're going to talk about now again. So this is kind of the emphasis of this evening's session. Right, so let's talk about intra-organismal signals in animals that are conducive to growth and development. So again, whenever we say intra-organismal, we just mean that this is signaling within the organism, so from one part of the body to another. So again, the signaling molecule that we're going to talk about this evening is a hormone. However, there are other signaling molecules, but this is the predominant thing. And of course, there are many, many different kinds of hormones. So in this example, you can see that you have an individual, and this is their thyroid gland. And their thyroid gland is also an endocrine gland, meaning that it synthesizes and excretes hormones or secretes hormones. So yeah, that's their thyroid gland, and it's going to show you some of the pathways that it can take to elicit its response. So the first step in this would be that the hormone is made in the endocrine gland. In this example, it's the thyroid gland, but of course there are many other endocrine glands in the body. So it's making this hormone, and then it's going to secrete this hormone to the extracellular fluid. So for humans, the extracellular fluid is the bloodstream. So what would happen for us is the thyroid would make that hormone. You don't need to know which one it is. It's going to secrete that hormone into the bloodstream, and it's going to kind of travel throughout the bloodstream. And depending on if it's water soluble or lipid soluble, it'll either need a carrier protein or not, but we'll talk about that in a second as well. And then as it travels through the bloodstream, it's carried by the circulatory system. So it just follows the natural rhythm. And so that circulatory system is going to take the hormone to the appropriate cell, which we would call the target cell. And at the target cell, there's going to be a receptor protein either embedded in the membrane of the cell or within the cytoplasm of the cell, depending on the signaling molecule and whether it's hydrophobic or hydrophilic. And so that receptor molecule, basically the signal is going to bind there, and then it's going to initiate that signal transduction cascade that we talked about right here, right? So this is kind of that last step where the signal arrives at the receptor, and then you get through all those steps until you get your response. Yeah, so like I said before, there are other kinds of signaling molecules that are involved in intracellular signaling. And even when we're just talking about hormones, there are basically two separate classes of hormones. And depending on which hormone you're talking about, the pathway that the signal is going to take from the endocrine gland it's secreted from all the way to the target cell is going to be a little bit different. So there are hydrophobic signals, meaning that they are lipid soluble. I didn't even fix my typo from Tuesday, but I will before I upload it. So yeah, hydrophobic means that they're soluble in lipids, but that they are not soluble in water. And then there are also hydrophilic signals. Again, another typo, but that means that they're water soluble, but not lipid soluble. So depending on the type of signal, the signaling molecule and its molecular composition, the signaling pathway that it takes to get to its target cell is going to vary. And also where the receptor is that is made to bind to that signal is also going to vary. So yeah, let's look at two different scenarios. So here on the left, you have a water-soluble hormone being secreted by an endocrine gland. And then on the right, you have a lipid-soluble hormone being secreted by an endocrine gland. So let's start with the one on the left. So this is a water-soluble hormone, meaning it's hydrophilic. So this endocrine gland is going to secrete this hormone into the bloodstream. Now, because it's water-soluble, it can travel easily in the blood without needing a carrier protein or a transport protein, because that the water is very hydrophilic, that the blood is very it's very polar. So if this was a lipid soluble or hydrophobic hormone, it wouldn't be able to do that. It would require some other protein to take it across to protect it from that polar medium. But basically, yeah, so this water-soluble hormone can travel easily in the blood, but what happens is once it arrives to the target cell, it cannot cross through the plasma membrane or the cell membrane. Now, the reason for that is, of course, because of that phospholipid bilayer that we've come to know and love, and the fact that it is only in order to cross that phospholipid bilayer, the molecule has to be small and it has to be non-polar. But if we're talking about a polar or a water-soluble hormone, then of course it can't cross that plasma membrane. So instead, there is a receptor protein located inside of that membrane where it has a space that is optimally shaped for this hormone to bind to. And then once this happens, of course, you get your steps that we talked about like a million times, right? So the hormone is binding to the receptor, you get your transduction, you get your response, depending on whatever the signal was in the first place and which receptor we're talking about. So that's kind of the one that we're familiar with. We talked about it last semester and also this semester, so it shouldn't be too new. But this time we're going to talk also about lipid-soluble hormones and what their pathway looks like. So lipid-soluble hormones are soluble in lipids, but not soluble in water. So they're hydrophobic. So this means that when the endocrine gland is going to secrete this lipid-soluble hormone, it's not able to flow through the bloodstream very well, because it's hydrophobic and because the blood is very polar. So it requires a transport protein to carry it through the bloodstream in order to prevent some aggregation and hydrophobic interactions and such, right? So even though it needs a transport protein to carry it through the bloodstream, once it arrives to its target cell, because it's small and nonpolar, it's able to cross that plasma membrane of the cell. So because it's able to cross that plasma membrane, there is no need for the receptor that is made to bind to the signal to be embedded in the membrane. Instead, this receptor can be somewhere in the cytoplasm. So it's basically the same idea, except this time it's going to bind to a receptor somewhere past the membrane as opposed to inside of the membrane. And then that receptor, of course, is going to initiate that whole cascade that we talked about like 700 times. So yeah, that's kind of the difference between the two. And now we're going to talk a little bit, like a very, very generic scenario about the sensory pathway and how things that you can sense, so whichever of the five senses it is, there are different kinds of receptors anyways for senses. So there are like mechanoreceptors, which can detect mechanical stress. There are thermoreceptors, which can detect heat, and that's the one in this example. But the point is there are lots of these, and each of them are equipped with their appropriate response that can be induced by the brain once it detects that signal. So in this example, you have this guy right here, and he is, I'm just assuming it's a guy. I don't know why, I just said that. But basically, let's say he is for some reason touching this flame of this candle for no reason. And so you can imagine that this is probably unideal and not very fun and probably hurts a lot. So the reason why it hurts, of course, is because he has sensory receptors that are densely concentrated at the tip of his finger. So these sensory receptors can detect the fact that there is an immense amount of heat present, and they will send this signal across through these sensory neurons, right? So we talked about neurons before, but basically these are able to pass neurotransmitters and communicate with the brain to initiate a response in other parts of the body. But yeah, so the signal will travel through a sensory neuron, and then an interneuron, and then it can also pass through the brain. So to the brain, obviously, the brain is gonna read the signal. It's gonna recognize that that there's a problem, that there's heat there. So then it's going to kind of tell. This is very like generic and not super scientific, but that's because you don't really need to know it in that much detail. But the brain is going to tell these neurons again, it's going to send out a signal saying, move the hand away from the flame. So basically, it's going to go back down through the spinal cord and then head over to a motor neuron. These motor neurons are responsible for movement. So this motor neuron is going to communicate back to the hand, basically saying, move your hand away from the flame, you bozo. So he does that, and all is well, and that's basically how it works. But of course, there are so many details that I left out, and that is because, thank the Lord, you don't need to know them this year, so it's not a big deal. I think if you're in psych and if you take the MCAT, you need to know, but for this, no. So don't even worry about that. So we talked a lot about animals and humans and whatever, but what about plants? How do they get signals for growth and development, and what are those signals even called? So their signaling molecules are called phytohormones. So it's kind of like ours, where ours are called hormones, theirs are called phytohormones, and these can basically tell the plant when to germinate their seeds. So basically, there are kind of three general steps involved in this process, but the first thing is that the seeds need to be watered, and the reason for this is that watering the seeds allows the embryo to get access to a sufficient amount of GA, which stands for gibberellic acid. And gibberellic acid is a type of phytohormone that enhances several types of growth. So in order for the seed or the embryo to get enough of this, it needs to be watered. The seed needs to be watered. So the second step is that digestive enzymes digestive enzymes in the seed, particularly in the endosperm, things like alpha amylase, which break down carbohydrates, they'll break down some of the nutrients in that endosperm so that they can basically be used to nourish the embryo as it grows and develops. Because it's really difficult for the embryo to take in if they're really large polymers, so it's more efficient for them to be broken down into their monomeric substituents, and then the embryo can use those monomers of nutrients and sugar to use it basically as fuel for growth and development. So that's kind of how this works for plants. And some of the differences between phytohormones and hormones, so animals and plants, are the following. So basically in animals, we have a bunch of different kinds of hormones. Like we have so many specific kinds, whereas in plants, they don't have that many phytohormones. There are not that many different ones, right? So the reason, so I guess like what to go along this, basically, the fact that we have many different kinds of hormones means that each one is assigned a specific task in the body. Whereas with plants, because they have very few different kinds of phytohormones, each one has a more generic and like more broad assignment of tasks, if that makes any sense. So because we have a bunch of different kinds, we're able to specialize each hormone to perform something specific. Whereas with plants, because they have very few different kinds of phytohormones, each one of them kind of has like an overlapping task with the others, right? So they all do very generic responses as opposed to more specific ones. And so another difference is that our hormones are produced by endocrine glands for the most part, not always, but phytohormones are just made in general throughout the plant. It's not really specific to a certain area of the plant. And the last comparison point, I guess, is that for animals, our hormones communicate very minimally between each other. They, because of the fact that there are lots of different kinds and each one has a specific task, they don't really need to collaborate that much because each one already knows what it's going to do. It can only do basically one thing or a couple of things. So there's not really a need to communicate with other hormones. But with plants, because they have very few different kinds of phytohormones and each one has a task that overlaps with the others, they need to communicate really well so that they don't end up doing something twice or overdoing something or anything like that. Or maybe one of them leaves out a specific task. So they want to communicate with each other to make sure that they're getting all the things that they need to get done. Whereas with us, with our hormones, because each one already has its specific task, it's not really necessary that they communicate too often. So those are some of the differences, I guess the main ones. So now we're going to talk a little bit about organism to organism signals. So we already talked about signaling within one individual, whether that was an animal or a plant, but now we're going to talk about signaling between individuals. So this can happen between individuals of the same species or between individuals of different species. And depending on this, the result of that signal is going to be, like the response to that is going to be different. So some of the types of signals that are sent between organisms include visual signals, chemical signals like scents, touch signals, and then also auditory signals, and it all depends on the species. So depending on the species, they can release certain pheromones, whereas others can't, or maybe they're not detected as well with a certain species, whereas with another species, the pheromones are very, very important. So it all depends. You can imagine. So yeah, if the two individuals are from the same species, the signals that they're sending out could be for the purpose of mating or for planning like a really strategic survival plan, right? But if the two individuals are from different species, then they're probably sending signals for the purpose of survival. So for mutualistic or symbiotic relationships, for example, just like the fact that, well, do you remember when we talked about the fact that there are some fungi that live symbiotically with plants, and we said that those were the ascomycota, are they the ascomycota? I think so, yeah. So they would live basically symbiotically with the plants, and they would allow them to basically obtain a bunch more nutrients from the soil. So they enhance their nitrogen uptake, they enhance their carbon uptake, their just a phosphate uptake, everything from the soil. And then in return, of course, they got the products of photosynthesis. So this would be a mutualistic or symbiotic relationship. And of course, they're definitely sending signals between each other, right? The fungus is sending signals to the plant, and the plant is sending signals to the fungus. And that's all for the purpose of this collaboration. So yeah, that's just kind of outlining the two differences between whether signaling is happening between individuals in the same species or in different species. So this is, I think, the last-ish part, but now we're going to talk a little bit more about the details of plants and how they receive and process environmental signals. So to do this, to get signals from the environment, especially when it comes to detecting sunlight levels and the type of sunlight that they're receiving, they're going to use these signaling molecules called phytochromes. And these phytochromes are these photoreceptors that are able to sense light. So basically, this was kind of confusing for me in first year, to be honest. And I do remember going to office hours and asking my prof to give me like a very like insightful explanation because I was like, what the heck? And I know that some of my classmates also felt the same way. And I also know that last year for PAL, there were some people who were confused. So if this did confuse you, I hope this helps, but let me know if you need a better explanation even after this. But basically, with these phytochromes, they basically have two different configurations, right? So these are two different conformations, if you will. So This one is the phytochrome red position. It looks like bunny ears that are like separated, and this is the inactive phytochrome. Now, when the plant is exposed to high amounts of sunlight, then the phytochrome is going to switch to the phytochrome far red position, and this is the active position. And so it's going to again revert back when it gets dark again. So it goes from being in the phytochrome far red position, so being active, and then in the dark when there's not as much sunlight, it's going to revert back to the phytochrome red position where it's inactive. So that's basically what you need to know. And you also need to know that in the sunlight, I think I have it on this slide. Yeah. So basically, chlorophyll, like the pigments that you find in the leaves of plants, the ones that make it green, basically, they're able to absorb red light when the sun when they receive direct sunlight exposure. So when the sun is beaming down on the plant, they're going to absorb mostly red light. But when the plant is in the shade, or it's getting kind of secondhand light, if that makes any sense, like it's kind of competing with other plants and it's not getting as much sunlight and it's mostly shady, then they're going to absorb mostly far red light wavelengths of light. So I guess that's the difference here. So again, when the plant is receiving lots of red light, so it's in direct exposure of the sun, it's going to be in the active configuration. And then when it's receiving much less light, so perhaps in the darkness or maybe in the shade, it's going to receive mostly far red light. It's going to absorb mostly far red light. So what does that even mean? Like, what's the point of this? So basically, like I said before, there are two phytochrome positions. There's the phytochrome red position and the phytochrome far red position. So if the sun shines its light on the plant and it absorbs, the chlorophyll pigments absorb red light, then the concentration of phytochrome far red positions will increase. So you'll get more of these if the sun is shining. So this means that the plant is active. So that means that the plant is able to use its energy because it knows that it has lots of it and that the sunlight is abundant. So it's able to use this energy to perform tasks that are not necessary for its survival. So things like seed germination and flowering. Of course, regardless of whether the sunlight is present or not, for as long as it can, it's going to continue to do the tasks that actually are required for its survival. But if it recognizes through the use of these photoreceptors that the abundance of sunlight is high, then they're going to use that to their advantage to perpetuate certain tasks that aren't necessary for survival, but they are good for propagating the population, for example. And then the reverse is also true, right? So if the photoreceptors, basically if the plant is in a very shady area or if it's getting dark out, then the chlorophyll pigments in the leaves are absorbing mostly far red light. So this means that the concentration of phytochrome red is going to increase. So this guy is going to increase in concentration. You're going to have mostly this configuration. So this is the inactive configuration, and that means that the plant is going to be inactive. So this just means that the plant is not going to perform certain functions that are not required for its survival. It's still going to keep doing its cellular respiration and all the things that it actually needs for survival, but the things that it doesn't need, like seed germination and flowering, that are just nice to have but not necessary for the survival of the plant, it's just going to stop those because it recognizes that it's probably not going to have enough energy to use on that in addition to its necessary functions. So what is the point of all of this? Well, basically, plants can use these signals to carry out certain functions only when the resources are available. So they don't want to start doing a bunch of things that are not necessary for its survival if they don't even have enough resources to do that. And it also allows them to recognize when they're not getting enough light in that PAR region. So do you remember the PAR region was the photosynthetically available radiation region? So it's that series of wavelengths that the chlorophyll pigments are able to absorb. So yeah, whenever these phytochromes also allow for the plant to recognize whether it's getting enough light in that PAR region. And so if they're not getting enough light, it might be because of other competitors nearby. So then they can send another signal over to those apical meristems, and then those apical meristems can enable that vertical growth so that the plant grows taller and it's able to outcompete the neighboring plants and get access to more sunlight. So yeah, I do have a video here. So when I post this on D2L, you can watch it. It's just kind of a summary of what I just said, but I think it does go into a little bit more detail that you might not need to know. So it's just like, watch it if you want, but not necessary at all. And Oh, I think this is one last slide. So, yeah, so how do animals receive and process environmental signals? Well, basically, there are a lot of different features that allow them to sense things differently, and depending on the species, of course, they will have these different features and different combinations of the features, and all of that has to do with what their niche is. So, what is their way of life? What do they feed on? Where do they live? What is their habitat like? You know, all these things play a role in that. But basically, there are some organisms, some animals, that have certain features like compound eyes. So compound eyes are those really like freaky eyes that houseflies have. If you've ever looked at one kind of closely, you know how it has like those massive eyes and it looks like there's like, it kind of looks like a kaleidoscope. So basically those are called compound eyes and they lack quality. So the housefly, for example, can't see things in like HD, but they're able to sense movement super, super quickly. So that's really beneficial for houseflies because I don't think they care if they can see you in HD. They just care if you're running at them with a towel. So they want to be able to fly away in time, but they don't necessarily need to see if you look good that day, right? So this makes sense and it's advantageous for the survival of this particular type of animal. But other animals will have single lens eyes like us. So this allows for us, for example, to see detailed images, and then sensing movement is a lot slower than it is in compound eyes. But the thing is that this is actually beneficial for us because we care a lot more, even for survival purposes. We care about seeing detailed images. We want to see things in good quality, and the sense of movement doesn't need to be as rapid as it would for a housefly. And that's just genetically speaking more advantageous for us. We don't really require that really, really intense sense of movement because we can already sense like how we can kind of tell when something's coming at us or not. It's just a little bit slower than it would be for like a housefly with these compound eyes. And that's fine because, we don't really need that to as much of a degree. And then another thing, which is kind of weird, is that there are some animals that are able to detect non-visible electromagnetic waves. So this is actually quite rare, but there are some animals that can do this. And one notable example would be certain snakes. So certain snakes can detect mice as they run below the ground. So let's say the snake is like slithering on the dirt, and then somewhere underground there are a bunch of like mice, or there's like one mouse, for example, and it's kind of running around under the dirt, if that even makes any sense. So it's really weird because the snake can't see it, the snake can't smell it, the snake can't really detect it, except for the fact that that mouse under the dirt is emitting very, very small amounts of heat. Obviously, like from like body heat, just like everyone else emits body heat too. But we would obviously never be able to detect that. Basically, nothing can. But for some reason, some snakes are actually able to detect the small amount of body heat that's being radiated in the form of electromagnetic waves by that mouse. So they can kind of follow the path of the mouse as it runs around based on following the electromagnetic waves due to the body heat that it's emitting. And then let's say the mouse is going to pop up out of the dirt to go above ground and like run somewhere else. The snake already knows exactly where it is and it's waiting. It's waiting for it to pop out so it can chomp and then have its dinner. So yeah, I think this was our last slide. I told you guys it was going to be pretty short, but yeah, so does anyone have any questions for me about anything? I'll stop sharing my screen. It can be about this stuff. It can be about something else. I do not care. I don't even think anybody's typing. Yeah, I don't think anyone's typing. So I guess if there are no questions or if you'd rather ask me privately, message me on Teams. I'm so much better at answering there than I am on email because I get a bunch of really random spam because I use my UNB email for random promotion stuff. Never do that. It's actually such a bad idea. You'll regret it. But yeah, have a great weekend almost, right? And enjoy the fact that spring is coming, summer's coming actually, and you're basically done with your first year, which is so good. And I don't know, you should be really proud of yourselves because it sucks. Like, I know it really does suck. I'm also like online and I recognize that I am so tired of being on the computer, but you're almost done. You're almost free from your shackles, and hopefully next year is a better year. But I am very, very proud of you for how far you've come so far because I can't imagine my first year being virtual and what I would do. So good on you guys. Okay, I'm done being the mom now. So if anyone has any questions, just message me and I'll see you guys next week. Bye.